My name is John Walsh. I'm a research leader in the School of Life Sciences at the University of Warwick. Within my research group, our research is focused on virus diseases of plants. We're particularly interested in virus diseases of brassica plants. That's things like oilseed rape, but also the vegetable brassicas, things like broccoli, cabbage, cauliflower. And our major interest in working with these viruses is to try and stop plants getting infected by them. And we're actually looking at things like natural plant resistance to the viruses.
I'm Dr. Max Newbert. I'm part of the Virus Plant Interactions group here at the Warwick University at the Wellesbourne campus. Many things can affect food production, such as disease and pests, but we mostly focus on disease, one of them being called turnip yellows virus. And this virus can actually reduce yield in oilseed rape by up to 46%, and in vegetable brassicas as high as 65%, vegetable brassicas being things like turnips and cabbages. The economic impact this can have can obviously cost the industry a large amount of money, which can affect things like food security and also affect farmers and productivity in the UK. Oilseed rape is a very important crop because not only is it an important foodstuff, but it's also used in the crop rotation. We have very high incidence of turnip yellows both in the UK and Europe, mainly due to its vector, what it's transmitted by, the aphid or greenfly. It has above 90% efficiency of transmitting the virus, and we found that around 80% of aphids can carry turnip yellows virus. Once a greenfly acquires turnip yellows virus, it actually has it for the rest of its life and able to transmit it to whatever plant it feeds on. Not only are aphids very readily able to spread the virus, but many weeds are reservoirs to the virus, so not only do we find it in crops, but we find it in species like dandelion or nettles that you might have all over the country, so they are a source from where these aphids can feed and then transmit the virus into crops. One of the struggles farmers have to actually identify their crops being infected with turnip yellows is that it has very muted symptoms, so it's very difficult to tell what's just an aging plant with what's actually an infected plant.
The big problem with this disease, or many virus diseases, is that farmers can't control the greenfly that spread these viruses around. Insecticides have been used, but we've seen crops that have been treated and have been 100% infected by the virus. And also a number of these insecticides are now being banned.
The other problems with actually trying to deal with the vector or the greenfly instead of the virus itself is that greenfly readily become resistant to insecticides, and because the lack of their effectiveness and new chemistry being very slow to come to market, it means that it's very difficult to control the virus through the aphids rather than the virus itself. So what we see is that natural plant resistance offers the benefit of reduced chemical inputs into farming, which obviously benefit for ecology as well as the farmer costs. And then obviously we're going to increase those yields by around 30% on average. So we see that natural plant resistance is the best way to go. So our team has discovered 3 sources of resistance to turnip yellows virus, one in Brassica apis or oilseed rape, and then again in closely related species, Brassica ouracea, which are things like cabbages, and Brassica rapa, which are plants like turnips. So we did a lot of work to understand turnip yellows itself, so we tried to find out what different genetic groups or different types of turnip yellows there was, so that we could test our resistant lines against each of those different types of virus to make sure that the resistance we found was durable against each, so that we had full resistance. We've taken that forward and we've started to cross our lines with commercial varieties to produce plant lines that will be usable for industry so that we can actually introduce this resistance into the field and increase those yields. Not only does our research aim to produce these resistant lines, but also to produce things called markers, which are used to locate the genes of resistance within each of our plant lines, which will make it much faster for us to breed it into the commercial lines.
Our work is an example of university research that's been publicly funded, and it's going to have a benefit for the general public and in terms of the outcomes of these virus-resistant crops. If you want any further information, you can have a look at our website or contact us via my email.